Hey everyone, welcome to Medical Microbiology. Today we will discuss chapter 12, which is the drugs or chemotherapy, microbe and host, the elements of chemotherapy. There will be 5 lectures over this chapter. The first lecture will be about the introduction to the chemotherapy therapeutic agents, and also we're going to talk a little bit about their targets. Okay, so these are the terminologies of chemotherapy. So chemotherapeutic agent, okay, chemotherapeutic agents are any chemicals that use in the treatment, relief, or prophylaxis of disease. So it's mean that we use this chemotherapeutic agent or drugs, or sometimes we call also medicine, whenever the disease or the pathogens already inside our body. So this is used for inside our body. Okay, compared to chemical, physical agents that use for controlling the pathogenic bacteria or microbes outside our body. We already talked about that in chapter 11, which is prevention. This one is curation, which is we try to cure our body from disease that already inside our body. Okay, so this is the chemotherapeutic agents or drugs. And prophylaxis, this is a drugs that actually used as a prevention. So we take this drug, we take this medicine to prevent us against the disease. An easy example is Tamiflu. Okay, so Tamiflu is actually a drug that have both function. It can be used for curations of flu, okay, H1N1 if you have that flu, but it is also can be taken by the family member that do not have the flu, but try to protect from getting the flu from one of the family member that has flu. So this is what we call the prophylaxis, which is the medicine or drug that is used for preventions against some disease. Antimicrobial chemotherapy, this is a general term for any drugs that we use for microbe. Okay, so this is not for special type of disease. Okay, this is for the terminology for drugs that used for microbes. Okay, especially pathogenic microbes. And then so over here we have the antimicrobials again, which is the ALS, the inclusive term for any antimicrobial drugs, okay, regardless of the origin. Okay, so this is we use this as a general term for any drug that uh, we take when we have the disease. Now, for the antibiotics, this is a very specific term. Okay, this is only actually used for bacteria. So when we talk about antibiotic, it is actually only used for bacterial infections. So we cannot use antibiotics for curing the viral disease, okay, like flu, for example. So we do not take the antibiotics for flu because antibiotic is for bacteria. And the semi-synthetic drugs, this is combination between natural and made drugs in the lab, okay, natural, and the drug that is chemically modified in the lab, pharmaceutical lab. Synthetic drugs, this is the drug that modified in the laboratory, okay? Pharmaceutical factory or laboratory. Narrow spectrum drugs, this is used for the drugs or the medicines that is only used for specific infection. Okay, specific infection, for example, maybe only used for gram-positive bacteria. For the broad spectrum, it means that this is the drug that can be used for wide variety of infection. Okay, maybe gram-positive and also gram-negative bacteria. Now, this is very important, for using the drugs. The drugs should be used for destroying the infective agents or pathogenic microbes without harming, so without harming the host cell or human cell. That's the primary principles of creating the drugs or using the drug for curations of disease or infectious disease. As it is, this is the ideal, this is the ideal characteristic of the antimicrobial drugs. First, which is it has to be, selectively toxic to the microbe only. Okay, it will not, it's not toxic to the host cell. It's not toxic to human cell. That's the primary or the first principle. The drug has to kill the microbes without killing our own cell. It has to be microbicidal. Remember, cide means kill. Okay, so the drug should kill the pathogenic bacteria or microbes rather than biostatic, rather only prevention of microbes, microbial reproduction. Because if it is microbiostatic, then this bacteria will be able to grow again, after several weeks of the drugs use. So it has to be the drug that able to kill or destroy the bacteria or the microbes. Relatively soluble because our body, is mostly consists of water. So these drugs should be soluble in our fluid, interstitial fluid or fluid inside our body. And then remain potent for long enough to act against that bacteria. It means that that drug will not be metabolized very fast in our body. Then it will lose its activity against the pathogenic microbes. Does not lead to development of antimicrobial resistance. So the drug that is used is not actually making the microbes become stronger. So we try to avoid this type of drug that causing the bacteria or the microbes to be stronger. And the drug should be complements. It is not the only chemicals that really kill the bacteria, but this is a complement or helper for our host defense. It means that our host defense still have to process the pathogens, the microbes, in addition to the drugs that being used. Remain active in the body tissue and the fluid, okay, readily delivered to the site of infection, reasonably priced, okay, does not disturb the host health by causing allergy or this is what we call the side effects. Okay, so as much as possible, it has very low side effects to the host cell. So this is ideal. It means that sometimes it is very difficult to fulfill these requirements. For example, there are some drugs that is very effective, but actually that drug can also causing side effects to the host cell. Okay, so origin the antibiotics. So when we talk about antibiotics, this is only used for bacteria. Okay, so antibiotics, there are many of antibiotics actually made in nature. So this is a natural antibiotics. For example, they produced by some microorganism. For example, some bacteria is actually produce the antibiotics. For example, streptomyces, they produce, a drug or antibiotic is called the streptomycin. Bacillus, maybe they produce other type of drug or antibiotic like bacitracin, for example. This is bacteria. Fungi or molds, in the genera of penicillium, it produce penicillin. Antibiotics called the penicillin. Cephalosporium, this is another fungi. So they produce antibiotics called the cephalosporin. So drugs, the antibiotics can be produced in nature by some microorganism. So they actually produce this for compete with other bacteria or with other microorganism. Like penicillium, for example, it's produced penicillin in order to destroy some bacteria that may be around them. So this is kind of competition. Okay, so they produce to kill other microorganism, and then we actually harvest these antibiotics, and then we use in our body to kill the bacterial infection. And again, this is the primary principle for development of drugs or chemotherapeutic drug, which is it has to be selectively toxic. Okay, for the bacteria, maybe it's easier because bacteria cell is, different than human cell. So the target that is used, for the medicines is going to be easier to be used for bacteria, okay, because they have different conditions, different type of cell compared to the human body. But it's going to be more difficult, okay, for infection that is caused by microorganism that is very similar to human cell. Like, for example, if it is caused by fungi, okay, protozoa, worm, for example, it's going to be more difficult to find the selectively toxic drugs because why? Because these cells are similar, okay? So they are similar to human cell. because they are eukaryotic cell. Right? So it's going to be more difficult to develop drugs for this type of infections, okay, fungal infection, protozoal infection, and worm infection compared to bacteria over here. So that's the reason why you will find that there will be more antibiotics or drugs used for bacterial infection compared to the drugs that used for the fungi or protozoa or worm infection. Okay, because it's not easy to develop selectively toxic drugs if it is used for fungi, protozoa, or worm. So again, about narrow spectrum and broad spectrum. Narrow spectrum mean a drug that only be used for certain type of infection, maybe for gram-positive bacterial infection only. But for the broad spectrum, this is a drug that can be used for many type of infection, including gram-positive and also gram-negative bacterial infection. Now the target, okay, remember when we discuss chapter 11, we also talk about the target for physical and mechanical agents on the on the microbes. Okay, so it's gonna be the same. It's gonna be the same. So again, when we talk about bacteria, bacteria have cell wall made of peptidoglycan, and then bacteria has cell membrane, okay, made of phospholipid bilayers, and bacteria has the DNA or chromosome, and bacteria also have the ribosome. Okay, so this is cell wall. Then under the cell wall, this is cell membrane. Okay, and this is DNA. Okay, and this is the ribosome. Okay, so remember ribosome is used for protein synthesis to make proteins, including enzyme. So bacteria are able to produce their own enzyme that is beneficial for themselves. Now, this structure will become the target for the drug. So this drug will have this target on this structure. So first target will be the cell wall, okay, inhibition of cell wall synthesis or destruction of the cell wall. Then the second target will be the cell membranes, okay, destruction of the cell membrane, for example. And the next target will be the DNA, okay, which is the nucleic acid synthesis, structure, and function of this DNA. And then the next target will be the inhibition of this process over here, okay, protein synthesis. So there are some drugs that will, destroy or block the protein synthesis inside the bacterial cell. And the last target will be the metabolic pathway. So these bacteria, they also perform metabolism. So there are some drugs that will block that process, the key process of metabolic pathway inside the bacterial cell. So these become the target for drug. So there are, for example, some drugs that only target the cell wall. There are some drugs that only target the cell membrane, nucleic acid, protein synthesis, or maybe the metabolic pathway. We're going to see that type of drugs over here. So this is kind of summary and information about several drugs that is used on bacteria, and they have special target. Like for example, penicillin, cephalosporin, vancomycin, bacitracin, carbaphinams, fosfomycin, cycloserine, isoniazid. Okay? These drugs or medicine, they actually destroy this cell wall, right? So blocking the cell wall. So when the cell wall is broken, then this bacteria will be die. Okay, so this is the what we call the cell wall inhibitors. The second is going to be the cell membranes inhibitor or destroyers. Example is polymyxin. So polymyxin over here will destroy the cell membrane. The next drugs will be the one that target to the DNA or RNA. Okay, so this DNA. So, you know, this bacteria have DNA, and DNA then will be able to produce the messenger RNA, and this messenger RNA then will be used for making protein by using the ribosomes. So there are some drugs that actually block this process. Okay, so the nucleic acid synthesis. Examples is the quinones. So quinolones or quinone. Rifampin, for example, this is also used to block this DNA RNA synthesis. There is also drugs that has their target on the protein synthesis. Remember, protein is made by this ribosome, and ribosome actually has two subunits. Okay, the large subunit and the small subunit. So there are specific drugs that only work on the large subunit like chloramphenicol, erythromycin, clindamycin, and oxazolidinones. This only blocking the protein synthesis on the subunit, the large subunit. There are also some drugs that actually work in the smaller subunit. So when this subunit, one of the subunit is blocked, then the protein, like enzyme, will not be produced, and then the bacteria will die because they need the enzyme in order to grow or in order to reproduce. Example of drugs that block the smaller subunit is aminoglycoside, tetracycline, streptomycin, and amikacin. Right? So these are the drugs that block the protein synthesis on the smaller subunit. And the last one will be the drugs that actually block the metabolic pathway. Right? So like, for example, bacteria able to produce a product from the substrate. So this required process over here. So there are some drugs that actually block this process. So when the process is blocked, then there will be no product. If there is no product, then the bacteria will die because they need this product in order to grow or in order to reproduce. Example of drugs that work in this blocking this metabolic pathway is the sulfa drug and trimethoprim. So this is just information about the drugs and the target, especially to bacteria. So we're talking about bacteria cells over here. What are the target for each specific drugs? Okay. Now, as we mentioned before, that the drugs that is used for bacteria usually have more diverse type because bacteria cells are different than human cell. So it's going to be easier to make the drugs that selectively toxic to bacteria only without causing problem to the host cell. Right? Because what? Because bacterial cell are different than human cell or the host cell. So if you see over here, there are so many drugs that actually used for bacterial infections. Eh? And then from here, you can also what is the target for each of these drugs, like penicillin, cephalosporin, bacitracin, aminoglycoside, for example. I mean the bacitracin over here, they all have the same target, which is the cell wall. Aminoglycoside, they use for RNA, eh? Protein synthesis. Okay, and then erythromycin, for example, the same is blocking the protein synthesis. Vancomycin also cell wall. Okay, the same like the penicillin. Tetracycline is gonna be protein synthesis. Chloramphenicol is the same, but remember they have different site. One is in the larger subunit, the other one is the smaller subunit of ribosome. Quinone, this is blocking the DNA and RNA synthesis. Okay, rifampin also RNA synthesis. Sulfra drug, this is the one that block the metabolic pathway. The same with the trimethoprim. So again, you will find more drug that used for bacterial infection compared to the infection that is caused by fungi, protozoa, or worms. Okay, over here, including viruses. Okay, why? Because these fungi, protozoa, and worms or helminths, their cell are very similar with the human cell. Okay, so this is a eukaryotic cell, right? So eukaryotic cell, so they are similar. Therefore, if we use the drugs targeting this cell, it might also targeting our own cell. So therefore, again, selectively toxic drugs that is made for this type of infection usually more difficult to make compared to drugs for bacterial infection. So you see less drug is used for fungi, protozoa, or helminth. Fungi, for example, amphotericins. This is for cell membranes. Okay, azoles for also cell membrane. Flucitocin. This is for inhibiting DNA, RNA synthesis. For protozoa like malaria, for example, quinones. Okay, or metronidazoles, helminths, bendazole diethylcarbamide, piperazine, niclosamide, ivermectin. This is for helminths or worm infection. Now, how about viruses? Viruses is not similar with our cell, right? So even viruses is actually not a cell. Why? There are less drug for viral infection compared to bacterial infection. The reason is because virus, when it is infecting our cell, it will enter into our cell. Compared to bacteria, usually they are outside the cell. Okay, so virus, they will enter into our cell. Remember the viral cycles? There are 6 cycles. Start with penetration. The attachment, penetration, uncoating, synthesis. After synthesis, there will be assembly and then release. So this process is happening inside the cell. So therefore, if we want to make a drug that kill this virus, most of the time this drug will also causing problem to the infected cell, to our own cell. So there will be more difficult to find the drugs for virus that is selectively toxic only to the virus without causing problem to our cell. Example for this viral infection or drug is the oseltamivir, cyclovir. This is mostly for herpes virus, for example. This is azithromycin or AZT. This is mostly for HIV infection. Okay, it's also known as AZT. So this is examples of drugs that's used for different type of infection. I think that's all for lecture number 1. So in lecture number 2, we will visit again the drugs and the target closely. So we're going to see which drug that actually destroy the cell wall, destroy the cell membranes, protein synthesis, DNA, and block the metabolic pathway. I think I see you on the next lecture. Goodbye now.